Okay, so we should be live on air now.
Mm-hmm.
All right, so welcome everyone to today's micro seminar, and I'm very proud to introduce you to our speaker, who is Dr. Tom Hanson. He's at University of Delaware, but he's like 80 miles away from me. He resides in our Biotechnology Institute in Newark. He's going to be telling us about his favorite bug, Chlorobaculum tepidum, which will be Chlorobium forever in my heart. But Tom had done his PhD at UC Davis, postdocs at Ohio State, and has been at Delaware for the rest of his career. And I will let him take it away. Tom, just a reminder, if you're in the Hangout, I will shut off your mic and your video. We'll turn it back on afterwards to have questions get asked. You could also ask questions through Twitter at any time. All right, Tom, take it away.
Okay, and Jen, if you could just make sure that the screen share is working.
It'll come in a second. Yep, looks good.
Okay, great. So thank you everybody for joining me virtually. It will be, a little bit odd not hearing people laugh or, fail to laugh at my jokes. And I guess the nice thing about having an online seminar series is that given my current health status, there doesn't need to be a splash zone established for the first couple of rows. I've been fighting a pretty nasty cough the last week. At any rate, as Jen said, I am affiliated with primarily the College of Earth, Ocean, and Environment in Jen's program, actually within the School of Marine Science and Policy. But I sit up at this Delaware Biotechnology Institute, which is literally, I think, 3 turns and about 85 miles away. I also have affiliations with the Department of Biological Sciences, the Delaware Environmental Institute, and the Center for Bioinformatics and Computational Biology. So what I'm going to talk about today is some work that I've been doing recently on studying sulfur metabolism in Chlorobaculum tepidum. And by I really mean that, it's actually my collaborators and I just am the one that gets to be able to come up here and talk about it. So some of the work I'm going to talk about today are done by current students in the lab, Katie Kalis, who works on sulfur-regulated proteins. I'm going to talk a bit about sulfur transcriptomics that has been done by Jake Hilsinger. Dr. Vijay Raman and Dr. Brian Eddy sort of helped get us started down this path, and then I'm also going to mention some data from Emily Turk, who just is the most recent graduate who did some work on the sulfur globule proteome. And then my collaborator here at University of Delaware, who I should give all the credit to any of the nice pictures that were taken. They were pretty much all done in Clara's lab. And the majority of them by Cassie Monaka, who's now an assistant professor at Niagara University. I'd also like to thank Leela in the Kelvin Lee lab for helping out with the mass spectrometry, or really doing the mass spectrometry for us. And then a lot of the other images were assisted or provided by Dr. Chandran Sabanayagam or Debbie Powell in our bioimaging facility. And then, of course, we always want to thank the people. NSF has been incredibly generous and consistent for me over the years. This has also been supported by an internal fund called the University of Delaware Research Foundation Strategic Initiatives Program that funded Clara and I to get this collaboration started. And then lots of toys provided by things like the INBRI Network, the EPSCoR Network, CBCD, and again, the Delaware Environmental Institute. So here's an outline for my talk. What I'm going to be going over today is a little bit of a primer on microbial sulfur metabolism and why I think it's cool and why I study it and what I would like you to take away from it. I'll talk about growth of Chlorobaculum tepidum on elemental sulfur and sort of the first surprising finding there, which has already been published. So maybe some of you have read that and it's not surprising to you at all. I'm then going to talk about sulfur globules, dynamics and properties, and close up with some work on identifying new players in elemental sulfur metabolism, and then a few closing thoughts, presuming that I have time. Okay, so to begin at the beginning, this is a simplified microbiologist view of the global sulfur cycle from a classical microbial physiology textbook, Lengler et al., Biology of the Prokaryotes. If we start over here on the left, geological sources of sulfur to the atmosphere, primarily volcanism. You have transfer through biologics by extraction of sulfur from soil and mineral reserves that then get volatilized as organic sulfur compounds or hydrogen sulfide. Obviously, industrial sources have become increasingly important over the last couple of centuries. This atmospheric pool is then re-equilibrated with these terrestrial and marine sources through precipitation or deposition, both over land and over the oceans. And ultimately, sulfur ends up getting cycled back through the lithosphere through tectonic activity and exchanging that way. This is really a quite simplified view and is a very sort of satellite level overview. This is much more realistic of what happens in situ. And this is an example that was provided by a collaborator of Jen and I's here, a colleague of ours here at the University of Delaware, George Luther. What this really depicts is what's going on in salt marshes. You have a lot of reduced and oxidized species, both inorganic and organic, that are constantly cycling and interchanging among different pools. And all of these species can overlap and be dynamically interacting with one another over millimeter size scales. So this is particularly true in marine and rich marine environments like estuaries and salt marshes. I won't talk about it today, but we've been doing a fair bit of work now on contributions of chlorobi to the sulfur cycle in the Chesapeake Bay. If you're interested in that, I can talk to you about that offline or at some other time. So sulfur cycling is pretty significant. It's actually one of the few, or maybe one of the increasingly recognized microbial activities that you can see from space. So this is an image off of the west coast of southern Africa. This is the Namib Desert over here. And this was a NASA satellite image. If we were to see this off the southeastern coast of England, we'd say that this would be a coccolithophore bloom. But as it turns out, and as was documented in this Nature publication, this is actually an elemental sulfur bloom that's caused by the oxidation of hydrogen sulfide, largely by chemolithotrophs that are blooming in the hydrogen sulfide rich waters here that occur after sediment disturbance in this area. What happens here is not only this sort of manifestation, but basically, if you look over here, seabirds feed on floating dead marine organisms. The water column here is really uninhabitable because the oxygen levels are so low, and all of the lobsters basically crawl up onto the beach so that they can at least breathe, even if they can't really survive for a long period of time at that point. In terms of area of this particular sulfur bloom, this is Delaware actual size relative to this scale image. So Jen is sitting about down here in Lewis. I'm about up here in Newark, and this sulfur bloom would effectively cover about the same surface area, or about roughly 1,800 square miles of the state of Delaware. So I'm going to simplify the sulfur cycle a little bit by focusing on a few key intermediates and activities. This is the way that I generally like to think about it to orient you. Reduced sulfur species are down here at the bottom, hydrogen sulfide with a charge on the sulfur atom of -2. The most oxidized form that we typically think of is sulfate, where the charge on the sulfur atom is +6. This means that there's 8 electrons available if you oxidize hydrogen sulfide fully to sulfate. And organic sulfur, like cysteine and methionine, is how we find it in our cells and tissues. That's at the -2 state. We're going to spend a lot of time focusing on this intermediate state, elemental sulfur. And I've colored the transfers or reactions here by dint of the organisms that catalyze them. So white arrows are catalyzed by microbial and macrobial processes. So sulfate reduction occurs in plants to generate hydrogen sulfide that's then assimilated into biomass. Chemolithotrophic sulfate reducers also do this as a way of disposing electrons. What we're going to be talking about today are reactions that are catalyzed by the green arrows, and primarily here the oxidation of hydrogen sulfide to elemental sulfur, and then the oxidation of elemental sulfur to sulfate that are carried out in light-dependent reactions by members of the group of bacteria called the green sulfur bacteria. So to introduce our model system, as Jen already noted, most of us grew up knowing this organism as Chlorobium. It got renamed Chlorobaculum, and eventually that was the one that stuck in the literature, even though some of us were trying to lobby for Chlorobium by putting both names in the titles of our papers for a while. This is an organism that was collected originally from Travelodge Hot Spring in Rotorua, New Zealand. I do have a little bit of guilt about this because I'm not quite sure that those samples were collected legally initially, but it's been a model system that's been passed into culture collections for a long time now. Why do we work on it? Well, this is essentially photosynthetic E. coli. This organism can grow with about a 2-hour doubling time. It's a strict anaerobe. If there's oxygen around, it will not grow. It doesn't particularly die off under these conditions, but it does not grow. And it's a moderate thermophile. So this grows at about 46 to 47 degrees centigrade, which is a little bit warmer than I like my showers in the morning, but it certainly won't burn you if you have to reach in and get tubes out of the bottom of an incubator. This organism utilizes reduced sulfur compounds, and it oxidizes them to provide electrons for autotrophic growth. The nice thing about it, it's got a relatively compact genome, roughly about 2,000 open reading frames that was sequenced by the Institute for Genomic Research and DOE. And as a postdoc, I, in parallel with other groups like Don Bryant's lab at Penn State University, developed a series of genetic techniques, including targeted gene replacement. One of the first things I did at University of Delaware was in vitro transposition mutagenesis. We can now make targeted epitope tags, and I won't talk about the mechanism today, but we're working on a paper to describe a now markerless mutagenesis technique where we can make mutants that are precise deletions or modifications of the genome sequence of this organism that does not leave behind any antibiotic resistance cartridge. So here are the basics about this organism. As I mentioned before, it utilizes these compounds as electron donors. Now, the most reduced of these compounds, hydrogen sulfide, these electrons come out of hydrogen sulfide with a redox potential of about -270 millivolts. That's not sufficient to actually reduce CO2 to the level of biomass. So these organisms have to use light energy to further drive the redox potential of these electrons negative to about -570 to -600 millivolts, where they can be put onto ferredoxin that drives the direct reduction of CO2 and assimilation via the reductive TCA cycle. Now, if you provide Chlorobium with hydrogen sulfide and thiosulfate, it actually displays taste. What it will do is it will actually oxidize the hydrogen sulfide first, and it produces elemental sulfur, followed by the oxidation of elemental sulfur. And eventually, once elemental sulfur is gone, it'll oxidize the thiosulfate, and eventually you do see sulfate as a terminal oxidized product of sulfur metabolism. One of the things that we were actually able to take advantage of is during the course of making in vitro transposition mutagenesis mutants, we collected this particular one, which we ended up calling C3 because of where it was found in our screen. And we noticed early on that this mutant accumulated this stuff at the bottom of flasks. This is actually a 125 ml serum bottle, and it turns out that this is elemental sulfur. This mutant has a defect that means that it's no longer able to oxidize biogenic elemental sulfur. And we had been struggling for a long time trying to grow trying to grow C. tepidum on elemental sulfur. We can't get it to grow on highly purified commercial sulfur. So what we ended up doing was purifying the sulfur from this mutant strain, feeding it back to the wild type, and lo and behold, the strain then grew, which then got us curious about why does it actually have to have biogenic sulfur in order to actually grow on elemental sulfur. So here's the experiment that details that initial observation and a few other points. This is a typical growth yield that we get because Chlorobium, when it's growing on sulfide, makes elemental sulfur. That means that we can't really use optical density as a great proxy for growth. So we tend to measure our biomass as micrograms of protein per mil, and that's what we do our growth yields in. If you take cells and this is my little cartoon for a C. tepidum cell, and you mix them with biogenic elemental sulfur, you get reasonably robust growth. The yield here is about 40 grams of protein per mole of sulfur oxidized, or 6.5 grams of protein per mole of electron utilized. And what that means is that the yield on this is actually about the same mole per mole as either on sulfide or thiosulfate. The reason this yield here is higher is because there's an awful lot of thiosulfate in this medium. There's a relatively low amount of thiosulfur, or of elemental sulfur, in this particular batch of medium. So we wanted to know a little bit about how this was actually occurring, because elemental sulfur is canonically thought of as being insoluble. It's not really truly insoluble, but it's definitely sparingly soluble at about 7 micrograms of sulfur per liter. So if we do an experiment where we put dialysis membrane into the same anaerobic culture tube, but we put cells and sulfur on the same side of the dialysis membrane, we get exactly the same growth yield. There's no statistical difference between these two growth yields. However, if we put elemental sulfur inside the dialysis and cells on the outside, or vice versa, we get no growth. We get no propagation of the cells beyond what we inoculated them at in medium with no electron donor added. I had really hoped that this experiment was going to work differently, because I had this really nice model where we had a shuttle compound that was going out and donating electrons to elemental sulfur, and then shuttling either polysulfides or organosulfur back to the cells. But this, in this case, was done with 50 kilodalton molecular weight cutoff. We've done it everywhere from 1 kilodalton up to 100 kilodaltons. It really says that a small soluble compound is not mediating any transfer between cells and elemental sulfur. And again, just to drive home the point, we've never been able to grow this organism on abiotic sulfur as the sole electron donor. Okay. So that's a little bit on growth of C. tepidum and S0. I now want to talk about sulfur globules, dynamics, and properties. So these are time-lapse microscopy images. They say that a picture is worth 1,000 words, but you know, time-lapse phase contrast microscopy movies might be worth 1,000,000 words, because it really enables you to understand what's going on. And again, this is really the major, result of the collaboration with Clara Chan's group. She has been an expert in this and really helped us understand and set up these experiments, which are not terribly trivial. So we have to keep these capillaries anaerobic at elevated temperature at 46, 47 degrees centigrade in light for, in these time lapses, these were run for about 12 to 16 hours. I think we've now gone up to about 24 hours where we've been able to look at cell growth. So on the left here, you see cells that are in an early stage of sulfide oxidation. And the first thing that you notice is that you see sulfur globules growing in from small sizes that are below the resolution, and eventually they come up and turn phase bright, which is a normal appearance for sulfur globules under phase contrast microscopy. The cells here are the dark oblong objects. And what you see is that while some sulfur globules do have at least transient contact with sulfur globules and cells, there are other sulfur globules that look like they've actually never contacted sulfur globules. And you see that these cells up here actually appear to divide quite happily without ever being in contact with a cell. So that may suggest that there are some soluble intermediates involved, or at least some below the resolution limit of this method compounds that are going out and populating these sulfur globules. This data has actually been published last year in microbiology. Now, if we take a look at what goes on during sulfur consumption, what we can see is actually very similar. You have here a sulfur globule that really never seems to be attached to a cell, but it decreases in size and goes away. You can see that cells that never make direct contact, or at least observable contact, with an elemental sulfur globule appear to grow and divide perfectly happily. And in fact, when we quantify the growth rates by looking at how fast cells are elongating in this condition, number one, by measuring that cell elongation rate, we were able to recapitulate the growth rate in our bulk cultures when we use protein as a biomass proxy. But also, we see no difference between cells that are growing and unattached versus cells that are growing and apparently attached to sulfur globules. So this actually, as a lot of good experiments do, brings up a lot more questions than it necessarily answers, but it does give us some new views on the perspective. The other thing that you notice here is that the C. tepidum cells are not static. They're moving, and we thought for a while that, okay, maybe this was thermodynamics or Brownian thermal motion, but it turns out that we've done experiments to show that this motion here requires both viable cells and it requires light. If you put cells in the dark at high temperature with no light, they don't move. If you put cells in the light at high temperature, but you've killed them before you put them in the capillary, they also don't move. So it seems like C. tepidum is actually modal under these conditions. So this was a bit of a head scratcher because C. tepidum had always been considered to be non-modal. So we went back, looked at the genome, and when we looked closely, we found that there is actually an operon or a gene cluster in the genome that encodes for 4 distinct but very closely related pillin subunits, and these are the flip-FAP type of pillin, which is a tight adherence pilus of gram-negative bacteria that's involved in twitching motility across surfaces. In this case, the pilus gets shot out in front of the organism, attaches to a surface, and then ATPases inside the cell retract and basically pull the cell along the pilus. We've taken a look at wild-type Chlorobaculum tepidum in negative stain electron microscopy, and if you can see these very thin white lines that are going across the image here, those filaments are actually of the right size that's been observed for tight adherence pili in other organisms. So we think that this has a functional tight adherence twitching motility. Whether or not it's involved in sulfur metabolism, we don't know just yet, but I'm working on overexpressing this entire genome segment as well as making relevant knockouts to assess the contributions of the individual genes here. The other note about this is that these genes look like they don't originate from the Chloroviaceae. All of the homologs within this gene cluster appear to have originated in beta proteobacteria, and probably as a result of a transposition, as we see at least 3 distinct copies, partial copies of transposase elements right next door. The other thing that we've done to try and understand how sulfur metabolism is really working in this organism is to try and take a look at the physical properties of elemental sulfur globules. Now, biogenic elemental sulfur has always been described of as liquid and amorphous sulfur. But in reading the literature, that's a little bit of an unsatisfying definition because nobody really says necessarily what that means. One of the things that we've done in collaboration with Chandran at DBI, and again, this was really done between Cassie and Chandran, was to take a look at atomic force microscopy as a way of interrogating the physical properties of elemental sulfur globules. So this is called a nanoindentation experiment, where this is a force curve that's generated by pushing the AFM tip down onto a sulfur globule. And you'll note that you have no force required here on the indentation. And then you get a pretty steady slope once you encounter the sulfur globule. Eventually, you get down to the substrate and it becomes much, much harder. This indicates that elemental sulfur is soft. And the fact that there's no real strong deviation here means that it is pretty amorphous. There's no real hard impediment to movement through this. It's not moving through liquid, but it's not trying to break through individual solid particles. The retraction curve looks somewhat similar. It's not terribly jagged. And you can do this over and over again. And in fact, if you do this 30 times in a row on more or less the same spot, you see essentially the same pattern. And in fact, if you image the sulfur globules after the first nanoindentation and the 30th nanoindentation, they look the same. There's no damage to the sulfur globule. In studies where this type of abuse has been levied against viral capsids or bacterial cells, you see surface damage. So this means that not only is the elemental sulfur sort of soft, it's also resilient. It does seem to rebound, or the way that I like to put it is that at least Chlorobium's biogenic sulfur looks like it might be the biological equivalent of flubber. I know that Jen really likes Disney references, so that's going to be my one Disney reference, Disney movie reference in today's talk. Now, it also turns out that, as I told you at the outset of the talk, that we initially purified our elemental sulfur globules from a mutant strain that can't oxidize elemental sulfur anymore. And we wondered if there were differences in how sulfur globules are consumed between this mutant sulfur, which is in this top strip here, and wild type sulfur, which are images in the bottom here. So the mutant sulfur tends to be quite large and fairly spherical, but it does have some surface textures in cryo scanning electron microscopy. This was published in the initial paper on sulfur metabolism back in at least electronically 2015. The paper date on this is 2016. You can see that Chlorobium cells do attach to this. And when Clara first showed this image, she really came down the hall saying, they're taking bites out of the sulfur. And this, it turns out, really appears to be true for the mutant form, this C3 mutant strain sulfur globule, and this is what we call a corrosion texture. If you look at cells that produce and make their own sulfur, what you can see is, and during the sulfur production stage of a culture, you see nice, again, smooth, sort of spherical sulfur globules. They don't necessarily need to be attached to cells, but you can find them attached to cells. And as you look at over time, you end up seeing that these end up going away, but you have little bits here that appear to be left over. And we call these sulfur globule remnants. And these, to me, look like deflated sacs. If we take a look at the physical properties of these sulfur globule remnants, we can image them in atomic force microscopy just like we did. We see something that's actually quite a bit different from the initial sulfur globules, and that is when you're moving through liquid, again, you see the same pattern, but when you're going through a sulfur globule remnant, you have a lot more structure and a lot more sort of variance in the resistance between different stages of both the indentation and the retraction, which says that there's a little bit more brittleness or internal structure in these particular remnants. So, taking you through what we've talked about so far, we've got really some contrary observations. We know from the initial data that I showed you, the dialysis culture experiment, that attachment really appears to be required for growth on elemental sulfur. And our static images show clear evidence for attachment. I didn't talk about it, but we already know that not all cells need to be attached, and that there has to be some role for soluble substrates providing for growth of unattached cells. And we published that along with the dialysis culture experiments in the first environmental microbiology paper. The time-lapse shows motility in Chlorobium tepidum, which was a big surprise, and it also infers that there's action at a distance, in that cells don't necessarily need to be attached to sulfur globules, or we can observe sulfur globules that apparently have never been contacted by cells throughout their history, either during production or during consumption. We now also know that the fate of elemental sulfur is different depending on whether it's been produced by the mutant. This tends to exhibit corrosion texture, whereas the wild type tends to leave behind these remnants. And these sulfur globules and remnants tend to have distinct properties. Now, we do know that if we age sulfur globules, if we take sulfur globules that we purified and we just leave them sit, we do see some aging and increase in the brittleness of this over time. I'm not going to talk about that in detail, but we're trying to understand both the mechanism and what that really means for sort of the ability of C. tepidum and potentially other organisms to utilize this as an electron donor for biological processes. So what I'm going to switch gears to now is focusing on composition and regulation to find likely players in sulfur globule metabolism. So we had known in the first paper that when we examined sulfur globules by secondary ion mass spectrometry, that the ions that we got off of the surface of sulfur globules were consistent with the presence of amino acids. And in fact, what I'd like to tell you in this section is that sulfur globules not only just have amino acids associated with them, it turns out that there appears to be a sulfur globule proteome. So if you take purified sulfur globules and you boil them in reducing and detergent conditions, you end up seeing an awful lot of proteins that you can resolve on an SDS page gel. If you try to sequentially extract elemental sulfur using just a ionic buffer, this is triethanolamine bicarbonate, this is a mass spectrometry compatible buffer, you don't see much coming off. You see maybe a little bit of this particular band coming out, but you don't generally get very high yields of protein. If you add a little bit of detergent, you get some protein coming off. If you add a lot of detergent, you get a lot of protein coming off. But in order to get to here, you really need to have both detergent and reductant. The interesting thing about this is that even though this isn't shown in this particular image, this SDS page profile is distinct from that of intact cells. And so we wondered what was the composition of the sulfur globule proteome relative to the encoded proteome of the C. tepidum genome. We'd already seen, again, by TOF-SIMS as well as TEM, EELS, SEM, EDX, some nano-IR data, and some just straight elemental analysis that there was evidence for carbon, nitrogen, and amino acids in the sulfur globules. In the earlier paper, we'd already identified these two proteins, which are only shared between the Chlorobaculum and Geobacter species that also interact with insoluble minerals in the environment. The interesting thing in our data is that the peptides that we've ID'd from these, at least for these particular proteins, are always smaller than we predicted from the genome, suggesting that they're going through some sort of processing before they end up in sulfur globules. So what else is there? This is work that was done as part of Emily Turk's thesis. Again, she just recently graduated, and we're looking at how to get this published. And if you look at the sulfur globule proteome relative to the bulk proteome, it turns out that the sulfur globule proteome is skewed. One interesting thing that goes up in the sulfur globule proteome is chlorosome proteins. This is the major antenna complex that's thought to be affiliated with the internal face of the cytoplasmic membrane. We have an increased degree of periplasmic proteins, both those that were initially annotated as periplasmic, as well as those that had to be manually annotated based on PSORT not necessarily doing the best job of this. Outer membrane proteins, we have an increase, we have a decrease in the number of extracellular proteins, or proteins that we expect to be exported to the extracellular space, and about the same number, maybe a little bit fewer, of cytoplasmic membrane and cytoplasmic proteins. There may be some evidence here for outer membrane vesicles. If we look inside of these, a lot of the proteins that we identify are what are called beta barrel outer membrane proteins, including CT1305, CT1804. This one is interesting. I'm going to come back to this and talk about it in just a second. This is an outer surface protein, but these are all structurally very similar to one another. in this large structurally classified family, as well as other outer membrane proteins, a predicted lipoprotein, or two predicted lipoproteins in CT1353 and CT2049. Coming back here to CT2144, we had previously published data on transcriptome sequencing for the transition between growth on thiosulfate and sulfide as an electron donor. We now have collected transcriptome sequencing data from elemental sulfur-grown cells. And what we see for this outer membrane protein encoded by CT2144 is that the messenger RNA is increased on hydrogen sulfide, and it appears to be decreased during growth on elemental sulfur as the sole electron donor. This is a structure of the CT2144 protein. This beta barrel cylindrical structure is what gives the family its name. The really interesting thing to us is that if you look at relatives of CT2144, you find that there appears to have been a really fairly dramatic expansion of this protein family within the Chlorobiaceae and perhaps within some delta proteobacteria as well. So we're still trying to understand the significance of this, and this is work that's really forming the basis of current students' work in the lab, Katie Kalis, who's working on this and a few other targets to really understand what their localization is and what role they may be playing in either sulfur globule formation or interactions with sulfur globules. The seed member for this family, LOM-R, is actually required for E. coli adhesion to hydrophobic surfaces and human cells. So, as that last slide probably is giving you a little bit of a flavor of, we know that gene regulation has to happen, and we know that gene regulation does happen. To take you back to where I started at with this, as a microbial physiologist, when I see patterns of temporal substrate utilization like this means to me that there have to be different biochemical systems expressed at different times that enable the cell to switch its substrate specificity. And we want to understand more details about how that's going on. And that's what a current student in my lab, Jake Hilsinger, is working on now. So in order to do this, to go beyond what we have in our transcriptome sequencing libraries, we're now utilizing a method called differential RNA sequencing, which is really a way of determining globally the transcript start sites across the genome of any organism of interest. The way that it works is that you harvest total RNA, and then you treat with two distinct enzymes. One is a TEX nuclease that specifically degrades messenger RNAs that have a degrades RNAs that have a 5' monophosphate. 5' monophosphates are typically enriched in processing sites or cleavage of RNAs. They are not the messenger RNAs. Messenger RNAs in prokaryotes typically start with a 5' triphosphate. What you then do after the TEX treatment is you take a phosphatase, trim that off so that you can ligate an adapter to this end of the RNA, and then use that to sequence. In the second library, you take the phosphatase and just do that treatment so that you're capturing the total RNA pool, both the monophosphates and the triphosphates. You then sequence these pools, align them to the genome, and what you typically see then is a TEX plus enrichment for 5' ends that have triphosphates, which again are thought to correspond to transcript start sites across a particular genome. And this was taken from the 2010 Nature paper that really laid out this technique. So if we do this in Chlorobaculum tepidum and we do this transcript start site analysis, we find that we get, as expected, several thousand transcript start sites, and we have a core of about 1,950 transcript start sites for, remember, about 2,228 individual open reading frames that we observe across all electron donors. There are substrate-specific transcript start sites for all three of the substrates, sulfide, thiosulfate, and S0, and we have ones that are shared between pairs of substrates. If we take a little bit of a closer look within these bulk pools, what Jake's found is that around 61% of the transcripts of primary transcript start sites have this motif, which really corresponds to a sigma 70 or housekeeping RPOD footprint. And this footprint is very close to, but somewhat distinct from that found in the most closely phylogenetically defined realm where this has been looked at, which is the Bacteroidetes. Another about 7.5% of primary transcript start sites have an RPOE, which is an extracellular stress sigma factor, still within the sigma 70 family of transcription regulators that are found in the Chlorobaculum tepidum genome. And primary transcript start sites are those that occur within 300 base pairs upstream of an annotation. Here's a couple of examples. Here's our mapped transcript start site, and here's where we're mapping the -5 and -33 boxes for our canonical RPOD motif. And you can see that across different genes, we have a fairly consistent spacing like you'd expect for this type of a motif. Now, to understand what's going on in terms of differential regulation, what we've gone through is paired our transcriptome sequencing results with the differential RNA sequencing results to try and build bins for motif identification. So what we saw on cells that were grown or that were growing on thiosulfate and then had sulfide added to them, we saw a dramatic downregulation of copy number for messenger RNAs for genes that are encoding proteins involved in thiosulfate oxidation, in periplasmic protein maturation, in the oxidation of downstream parts of the sulfur pathway in the cytoplasm, and the oxidation of elemental sulfur, as well as one of the sulfide quinone oxidoreductases. In contrast, we saw upregulation of this high concentration specific sulfide quinone oxidoreductase, a polysulfide reductase complex, and some potential regulators that I'll talk about in a moment. If we look at the ribosomal proteins, though, between the two growth conditions that we've looked at here, we don't see much differential regulation, which says that the regulation that we're seeing in these genes, or at least indicates to us that this is not growth rate dependent regulation, but that this is really substrate dependent regulation. So now with our transcriptomes from sulfide versus S0, we've done the same analysis, and we can see that the regulation pattern for sulfide is pretty similar, irrespective of whether you compare it to elemental sulfur versus thiosulfate. If we look at particular motifs associated with bins of these sequences, Jake has been able to identify a number of these. I'm only going to mention this one in particular. This motif here happens to be associated with this sulfide quinone oxidoreductase that goes down somewhat in expression when sulfide is added to cultures. It's associated with this sulfide quinone oxidoreductase that goes up, and it's also associated with CT1277, which is part of a 2-gene operon that appears to encode regulatory genes. In this case, CT1277 itself is a helix-turn-helix member of the xenobiotic response element family, the ZRC family. And what we've recently found out is that if we make a deletion mutant in Chlorobaculum tepidum that lacks CT1277, what we lose is the ability to repress CT1087. So it seems like there's an autoregulatory loop that's going on as well in between CT1277 that may involve the other part of this operon, CT1276, that's involved in repression of genes that are expressed at low levels under sulfide and that get induced when sulfide is added to cultures. So this is the mutant data for 2 different strains of the mutant for CT1087 messenger RNA data. And here is the result for when you add sulfide. Now, the interesting thing is we don't have the full story here because in the CT1277 mutant, we still get an increase in expression in the CT1277 mutant, meaning that there has to be transcriptional activation as well. This shot here just shows where this putative sulfur operator sequence is located in the CT1277 promoter sequence, the CT1087 promoter sequence, and CT0117. In general, it either overlaps either the transcript start site or elements of the sigma 70 binding motif. So with that, I'm going to conclude and wrap up and give some final thoughts here and a few closing ideas. So again, at sort of the outset of the second part of this talk, I said that there were some contrary observations. We know that attachment appears to be required for growth on elemental sulfur, and we've got evidence for both attachment being required and for action at a distance, which implies that there still has to be some room for soluble intermediates, or at least intermediates below the resolution at which we've looked at so far. And we know that sulfur globules that are freshly formed have distinct properties from remnants or even aged sulfur globules. So we think of freshly formed sulfur globules as being soft, pliable, amorphous, and tasty. As they age, they're still mostly soft, but they get some aged brittle domains. And then if you look in the remnants, basically, Chlorobium has sort of taken out all the good juicy bits, and it's littered, leaving behind the bits that it doesn't necessarily want to deal with. To me, what this may indicate is that there's got to be some large molecules or large complexes that are involved. We think maybe that pili, in terms of the tight adherence pili, or possibly outer membrane vesicles based on the sulfur globule proteome, may be involved, but we have not ruled out yet some soluble intermediates. And we've started really making some inroads on regulation and understanding the mechanism, or how Chlorobaculum tepidum is really making choices between what sets of genes it expresses based on signals that it's perceiving in its environment. So here's a synopsis. What do I think is happening? And I put this disclaimer here, is that, you know, I may be a lone mid-career potential crackpot, where my collaborators and students are probably begging me to stop. Dear God, please stop right about now. So what I think may be happening is that in sulfide oxidation, we know that the key enzymes for sulfide oxidation are found probably in the periplasm, and that means that the initial steps of sulfur formation are taking place in the periplasm. My current favored model is that sulfur is actually being exported from cells already coated, probably as an outer membrane vesicle, and that these small outer membrane vesicles end up traveling to, I'm not sure that diffusion is the right word here for what would really be a sort of a colloidal particle, and then end up aggregating through vesicle fusion into the large sulfur globules that we can see that are about cell size or even larger. Pili may be involved in trafficking outer membrane vesicles out to sulfur globules. We don't know, but we're working on genetic and observational experiments to really try and assess this much more concretely. In terms of sulfur consumption, this process has to be reversed. Do the cells send out new outer membrane vesicles that are now geared towards degradation of sulfur globules? Are pili involved in trafficking? We don't honestly know at this point. We do think that there's evidence probably for polysulfides being involved in eventually the cross-feeding of cells that are actively metabolizing elemental sulfur and producing remnants, and those that we saw microscopically that are growing at a distance from sulfur globules. So what were the surprises here? I referred to a few surprises in the title of my talk. Well, the first and biggest for me, was that C. tepidum is modal. If you look at the description in Burgess, these organisms are non-modal. We think we have a handle on why that is. Second is that extracellular sulfur does not deform under repeated abuse, unlike under other biological structures like cells and viral capsids. Third, C. tepidum is a litter bug. It leaves sulfur remnants behind, and if those sulfur remnants contain bits of the sulfur proteome that we find associated with freshly made sulfur globules, then that's, in terms of energetics, a really expensive littering habit. And it seems to me that there would have to be a really strong evolutionary reason for that, which I'm happy to speculate on during questions. And then finally, we're beginning to open some insights into sulfur-driven regulation. Ultimately, this is really cool. I'm fascinated by this system, but what does this actually mean for potentially larger questions? Well, one thing is, can we do something with this? this picture in the lower right is from Alberta in the oil sands region. This is elemental sulfur that has been stacked up. These images back here, those are 3- and 4-story buildings. These are about the size of football fields, and this is not the only site in Alberta where sulfur-rich tar sands oil is being processed and producing elemental sulfur that currently really has no sort of industrial utilization. You can use some of this for fertilizer. There's other people that are looking at whether or not this has industrial uses in sort of next generation smartphone batteries, but it's really early days. Well, if we can make this type of sulfur, which chlorobium doesn't really grow on right now, if we can take the insights that we've had from chlorobium tepidum and use this in sort of a chlorobium synthetic biology, we might actually be able to convert either hydrogen sulfide or these pyramids of elemental sulfur into value-added end products like ethanol, sulfuric acid, which has a lot of industrial uses, or to perhaps a more bioavailable form of elemental sulfur than this highly chemically purified stuff that's being made currently. This is some back of the envelope calculations that I've done where even at low efficiencies, these processes, which I would envision as polishing either natural gas, biogas, or flue gases, could offset about 4% of corn ethanol per year, even only operating at 10% efficiency. So if anybody from ARPA-E is listening, this is sitting at your doorstep, and if you want to get back on what's going to happen with that pre-application, that would be great. So finally, I'd like to make a plug. As some of you may have seen on Twitter, we now are in the stages of finalizing and launching a new graduate program in microbiology at UD, targeting a first full externally recruited cohort in 2018. It's going to involve about 40 faculty plus faculty across 5 out of the 7 colleges at the University of Delaware. We're going to have core courses in genetics and genomics, physiology and diversity, with the ability for faculty and students to affiliate across 4 distinct concentrations: environmental, host microbe interactions, applied microbiology, and then physiology and genetics. We've got a Twitter handle for the program, we've got a website, we've got a Facebook group, and if you're interested in more about this, please feel free to contact me. So with that, I'm actually pretty much spot on what I had planned for time, about 50 minutes. So if you like that, I'm perfectly happy to answer questions about that. And if you didn't like that, I'm perfectly happy to answer questions about anything else, ranging from work that we've done in tundra soil microbial ecology to nitrate reduction pathways to low molecular weight thiols.
All right, thanks, Tom. All right, and I liked how you said I was sitting in Lower Delaware. I walked about a mile and burned 200 calories while you're doing that.
Nah.
So if you want to shut off your screen sharing, we'll see if there's questions inside the Hangout. Anyone who would like to ask a question, feel free to buzz back in. I can turn you off mute if you need it.
Okay, yes, it looks like I am there.
Okay, we can see you now. So I guess I'll ask the first question while we're waiting for other people. Just ping me if I need to unmute myself. With the motility thing with the pili,
yep.
Right? How many other genomes is something like this seen in, and maybe there's more widespread motility than we realize due to twitching?
That's a really good question. I haven't looked at it. This particular one is fairly narrow. Where it's been looked at the most is, with most things in gamma and beta proteobacteria that are of pathogenic interest, because it turns out that the same tadpilli, like an Acinetobacter, are really important virulence determinants. And the seed for this particular, or the seed, one of the seeds for the pillin families is also involved in adherence of E. coli to intestinal epithelia. So it's really been biased. I don't know. I haven't looked at it in that much detail yet. I'm really focusing on trying to make sure that we've got the right set of genes by making the mutants, and also seeing if we can't make sort of a Bob Ross Chlorobaculum by overexpressing the pillin genes and making it really hairy, just because it would be cool.
Or give it a happy little tree, right?
That's right. Have it.
Okay.
Well, it would be a happy little tree because really it has photosystem 1 in it.
Yeah. There you go. All right. Anyone else? Could you say a little about whether or how universal this is among other phototrophic sulfur bacteria?
Right. So I did not explicitly make the point. So this is a really good question about how close, in general, sulfur oxidation pathways and mechanisms are between different groups of organisms. So the other group where it's really been studied most closely are the phototrophic sulfur bacteria that are proteobacteria. In that system, the sulfur globules are not extracellular. So the sulfur globule, in most cases, so in something like Allochromatium venosum, the sulfur globules are actually retained within the periplasm, and they seem to be coated with a coating of sulfur globule proteins. There appear to be three types that are all interrelated, SGP, A, B, and C. So that seems to be a mechanism by which the sulfur isn't sort of allowed to go out beyond the periplasm. For cells that do send sulfur out into the environment, it's really unclear whether or not this is going to be a common mechanism or not. So we do have strains of Ectophila retispira, which is a purple sulfur bacterium that actually does produce extracellular elemental sulfur globules. And yeah, we're playing around with that a little bit. In terms of the basic enzymatic machinery of how the electrons are gotten out of elemental sulfur, irrespective of where the sulfur is localized, there's actually fairly good congruence in between the DSR systems and things like that, between, again, the best studies in cremation and the studies that have been done by my group, by Freegard's group, and when he was a postdoc, and in Don Bryant's lab, and in his own position now at University of Copenhagen.
All right. So I don't see any other questions coming in. Anyone in the Hangout want to buzz in? Just let me know. If not, I will thank Tom for his seminar today. And we've got a full series headed out through the rest of the year. So be sure to tune in next month. So thank you, Tom.
Yep, no problem.